Hello, everyone, and welcome to ACS Webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Erin Girardi, and I'm pleased to be your host for today's broadcast, which is being co-produced with the ACS Reactions. We would love to get you involved in the discussion. You can share your thoughts and questions in the questions panel. You can also join the discussion on Twitter by including AmerChem Society or the hashtag ACS Webinars in your tweet. The slides from today's presentation can be accessed now in the Handouts section. An invitation email to view the unedited recording from today's presentation will be sent to all registrants. It will be open for a 24-hour period, after which it will be removed, edited, and will become an ACS member benefit. Our moderator today is Monica Mattesi, who's been a science educator for the past 14 years, both at the secondary and post-secondary level. Before she became a science educator, Monica's interests included performing autopsies and studying forensic science. Once she completed a BS and MS in forensic science, she established her career as a criminalist in the Controlled Substance Analysis section of the NYPD Police Laboratory. She began her teaching career a few years later in Harlem, New York. She spent 8 years in Harlem and at John Jay College of Criminal Justice before she began pursuing her PhD in science education at Stony Brook University. Monica is currently an education specialist and lab manager at the Institute of STEM Education at Stony Brook University. And with that, I'll turn it over to Monica to tell us about today's bloody topic and speaker.
Good afternoon, everyone. And on behalf of ACS, I'd like to welcome you to Bloodstains and Biomolecules, from crime scene to silver screen. Our speaker today is Dr. Theresa Stotesbury. Theresa Stotesbury is an assistant professor and early career researcher in the forensic science program in the Faculty of Science. Her research group characterizes how biological tissues, like blood, degrade in the environment and finds innovative ways to use this information to help with forensic investigations. For example, recently, her research group was highlighted on the front cover of the journal Forensic Chemistry for mass spectrometry imaging of fingerprints that were chemically removed from underneath bloodstains. The first study of its kind with significant implications for forensic investigations. Her group uses methods in analytical chemistry to develop models to age a bloodstain, which can be useful in providing context to the events of a crime. Her research group also focuses on developing novel biomaterials for biosensing and forensic science applications. Dr. Stotesbury has held a passion for forensic science since her earliest years as an academic, graduating with a Bachelor of Science from Trent University and a Master of Science from the University of Auckland in forensic science. In 2017, she graduated with a PhD from Trent University in material science, where she developed a synthetic blood substitute that forensic agencies around the world are now incorporating into their research and training practices. Dr. Stotesbury regularly collaborates with members of the forensic community and has held a previous appointment of research scientist at the Ontario Provincial Police. So now, before the talk, we're going to watch this great video that will help introduce the topic of today. And this video is going to give us a background on the chemistry of blood and its use in past and present films. So we're going to see the way different types of blood, or different types of ways that blood can be created for the silver screen and how it has evolved over time. And then we will begin our talk.
All right. Hi, everyone. My name is Theresa Stotesbury, and I'm really excited. Wow, ACS. Thank you so much for having me, and thanks for the introduction. So you may be wondering who is this Theresa Stotesbury, and where in the world is her research group? And while I'm not traversing the streets of LA and making fake blood for Hollywood, I am up in a university called Ontario Tech University, just north of Toronto. And before we begin my webinar today, I'd just like to let you know that at Ontario Tech University, we acknowledge the lands and the people of the Mississaugas of Scugog Island First Nation. We are thankful to be welcomed on these lands in friendship. The lands which we are situated on are covered under the Williams Treaty and the traditional territory of the Mississaugas, a branch of the Greater Anishinaabeg Nation, including the Algonquin, Ojibwe, Odawa, and Potawatomi. And these lands remain home to a number of Indigenous nations and people. And so I know people are checking in from around the world, so hello and welcome. And this is just a little bit about where my university is from and where some of the research that you're going to see comes from today, and some other places too. All right, so what am I here to do today? I'm here to break down some myths about fake blood and from the silver screen to the crime scene. Before we do that, we're going to look at forensic science in general and where fake blood can be useful in criminal investigations, mostly in research and training. So we're going to focus a little bit on bloodstain pattern analysis. Generally, what is it? What are some key things that BPAs in the field do? Can you see my screen? Hopefully, I'm just getting a pop-up and seeing my screen. Hopefully that's all right. And so, can everyone see me?
We
Can you hear
can
me?
see you. We cannot see your slides.
Oh no. Okay, that's too bad. Let me pop up. Can you see my slides now?
There we go.
Okie dokie. Let me go back. Apologies for that technical difficulty. My apologies. So here I am. Hello and welcome, and thank you so much for coming to my webinar today. So today we're going to look at bloodstain pattern analysis, more specifically where forensic blood substitutes are useful in research and training, and then we'll dig a little bit deeper on fake blood. And I'll pose the question, are fake bloods, is there one type of blood substitute that fits the jack of all trades, or do you think blood substitutes are created for a per use basis? And I'm sure you can hypothesize. In our forensic toolkit, we have a lot of different materials that are available to us to use in forensic research and training. I'll specifically go through two main types of materials that comes out of my research and my laboratory group's research. We'll look at hybrid inorganic organic sol gel materials, and then we'll look at briefly a bonus treat to the webinar today, some alginate-based hydrogel materials that our group is now turning into forensic tissue simulants. It's a really exciting research. Let's first look at the field of bloodstain pattern analysis. And I should mention too, in my talk, I do have some photos of bloodstains. There's not many of them, and most are animations. So if at any point the photos of bloodstains make you feel uncomfortable, please feel free to exit the webinar, and you can contact me or check out my group's papers for more details. Back to it. Let's look at bloodstain pattern analysis. So by definition, if you were to go get a textbook and look up what bloodstain pattern analysis is, or BPA as we call it, it's a field that is primarily focused on the study of the morphology of blood stains and bloodstain patterns. So it's the study of the size, the shape, and the distribution of blood stains. And the whole point of this is in order to determine the physical events in which gave rise to the origin. So how did the blood get to where it's being observed at the crime scene? Can you put that information around bloodshed into context in a criminal investigation? So by definition, we're looking a lot at morphological properties of bloodstains. But even before that, the bloodstain pattern analyst has a lot of chemical and biological tools in their toolkit in order to collect a wealth of information about blood at a crime scene. For example, a BPA might be called to a crime scene to observe what appears to be a bloodstain pattern say on a wall or on a surface or a substrate. The BPA in their toolkit, they have the ability to apply some chemical reagents in order to do some presumptive testing for blood. So in fact, this presumed red to test and hypothesize, is this red staining presumed to be blood or not? One way they can do this is say using a hemotrace card, for example, to test for human hemoglobin. If that happens and the red staining is presumed to be blood, they can also swab and collect the blood stains, and that can be sent to the lab for further processing, say for source identification and DNA profiling. Something I won't get to in today's talk, but you know, that's a very important take home from in terms of evidence that blood can give you. Now back to the meat and potatoes of the definition of BPA. So we're really looking at blood stains, their morphology, and now the BPA really gets to work. They can look at this pattern here and start hypothesizing what type of pattern they're observing. They can look at the stain size and shape and see some stains are sub-millimeter. We have some stains with diameters in the centimeters or not. Notice this fan-like distribution and directionality. If you see my little laser pointer arrow, you can see that there's kind of like a fan-like distribution and there's directional staining. In terms of its distribution. So they might start hypothesizing based on their training and what they know about the field, this is probably an impact pattern. Impact patterns, these are spatter patterns, note, not splatter, spatter, these are spatter patterns, and they're created as a result of an object striking liquid blood. And what's interesting and important about these types of patterns is that you can collect morphological data. You can use the shape, particularly the ellipses in these blood stains, carefully select specific blood stains and perform a three-dimensional analysis to figure out where in space was that blood source located at the time of bloodshed. Very important. Now, not quite yet in the practitioner's toolkit or the BPA's toolkit is the ability to actually age a bloodstain and say, oh yeah, for sure, it's 2 weeks old or 2 months old. But there is a collective body of research occurred over the past few decades that's looking at ways to chemically age or biologically age, use some sort of biomarker or biomolecule analysis in order to age a bloodstain. Okay, and so, I mean, if you just look visually, you can see that blood stains do change in color over time. They look for bright, fresh red from the initial point of deposition on a surface down to kind of like blacky brown, and this happens when blood stains are quite old. The rate of this change is dependent upon a lot of environmental conditions, and there's a lot of biomarkers that contribute to the degradation of a blood stain. Now, we saw from that YouTube video, we saw that heme was heme, or the hemoglobin, and particularly the heme in the hemoglobin is an excellent biomarker in order to show these changes, particularly these chromophoric shifts in the blood stains over time. And blood ex vivo, so when it's outside the body, it will undergo a series of oxidation mechanisms within the body. We see oxidation from deoxy to oxyhemoglobin. Sometimes we see some further oxidation to methemoglobin. However, that reduction is actually possible due to enzymatic reduction in the body. But in ex vivo conditions, when a blood stain is formed outside in the world, these enzymes aren't really available. So we see further oxidation, or a lot of the oxyhemoglobin will continue to oxidize to methemoglobin. And then we'll see protein, the protein will continue to denature and form these hemichrome and hemochromes. And really that just, their names are dependent upon the oxidation state of the iron. Okay, so, but I mean, generally, it's important at the end of the day for the BPA, when they're doing their analysis, writing their report, they're sticking to the scientific method and they're reporting on the limits of their knowledge. And you might ask, well, where do they actually obtain their knowledge from? Where are they going? How does one get into blood stain pattern analysis and forensic science? Well, BPAs around the world are typically trained using a series of courses and a series of tests, for example, in order to gain their expertise in the field. They'll take a variety of courses. In Canada here, for example, BPAs will take basic and advanced courses. They may be asked to take additional training, or they may take additional training in fluid dynamics, chemical enhancement, so understanding how and when it's appropriate to use protein versus peroxidase versus maybe amino acid staining reagents at crime scenes. There's additional studies like looking at morphology of bloodstains on complicated surfaces like fabrics, for example. And here in Canada, most of the BPAs undergo an understudy program where they shadow experts. Their reports are peer-reviewed, and simulation examples are peer-reviewed, and sometimes they do research. And so all of these training exercises and research opportunities all require some sort of handling of blood. And this is particularly true in an experiential learning context. But what happens, and believe me, it does happen when you can't source human blood. You can't source human blood for research and training. What are the options out there available to a BPA or to forensic agencies? And so if human blood can't be sourced for some sort of biohazard, maybe ethical or cultural reason, can other blood sources be used? The answer is yes. There's a significant body of research that looks and validates mammalian blood sources for bloodstain simulation in the forensic sciences. A lot of these blood sources do have the addition of anticoagulants, and there's a variety of literature out there on which anticoagulant to use at what concentration, depending upon the mammalian blood source. And sometimes scaling factors are needed, sometimes they're not. Perhaps that's not, perhaps mammalian blood can't be sourced for whatever reason. Can you use semi-synthetics? Sure, of course. And what do I mean by this? I mean, these are some sort of synthetic product that do contain biomolecules, like for example, purified hemoglobin. They demonstrate some nice physical properties and chemical reactivity properties too. And can you use anything else? Can you just go to the store and get the fake blood that you would maybe use for your Halloween costumes? Maybe, likely not, but there are a whole branch of synthetic materials that do exist. And these are materials that don't contain many biomolecules, if any at all. And again, out of all of these types of sources, if you can't use human blood, it's important to use some sort of fluid or some sort of material that's undergone some sort of validation for the research and training or case-specific details at hand. Okay, and so I thought before I go into talking to you exactly what forensic scientists care about, I thought I'd ask you. We saw a YouTube video and we saw some things that Hollywood is really excited about when making fake bloods, but I want to ask you, what makes a good forensic blood substitute for forensic research and training purposes?
Okay, so everyone, the poll is live, so get your votes in. You can vote for more than one. So what do you think makes a good forensic blood substitute? Visual appearance, oxygen transport, viscosity, wettability and other surface interactions, or something else? And if you're choosing something else, you can tell us what that is in the chat. So get your votes in. We will be closing the poll shortly. And also, if you have other questions for Theresa, please feel free to get them in the chat as well. We'll close it in 5, 4, 3, 2, and 1. Okay, so we had a tie for the most votes, both at 71%. People voted for viscosity and wettability and other surface interactions, followed closely at 65% with visual appearance, and then at 17% oxygen transport, and only 3% voted for something else.
Great, thanks so much. Well, all these are great answers, and it's important to note that for the most part, we care a lot about, why can't I see my mouse? There we go. We care about the physical and chemical properties relevant to forensic science. And so there are a lot of blood substitutes and clinical blood substitutes out there that primarily focus, say, on oxygen transport. While these blood substitutes, even red blood cell simulants and other cellular simulants, are fantastic. They're amazing. They're amazing materials, and the innovation is quite impressive. At the moment in our community, those particular materials don't actually meet a lot of the fluid properties that are important to the forensic scientists because fluid properties really dictate the morphology of the bloodstain on the surface. We care about visual appearance, we care about viscosity, we care about wettability and other surface interactions, and we care about some other things too. So let's go and look a little bit more in more detail. So the requirements and the reason why I say our forensic blood substitutes are not, we don't typically use fake blood from Hollywood, for example, in our research and training initiatives, because they don't quite meet the fluid properties and the relevant fluid properties for bloodstain simulation, and particularly in their morphology. Sure, they might look great, but we care a little bit. If you zoom in on a bloodstain, we care a lot about the fine detail on bloodstains. For example, we care a lot about the size of the bloodstain and also these fine details like spines and scallops. Depending upon how the spherical blood droplet impacts the surface, we'll see varying morphologies in terms of their shape. You can fit an ellipse around really the parent area or the elliptical area of a given stain. This is a type of drip stain on a surface, and this is what's useful for determining, say, things like the angle of impact, which can then be used in, say, impact, like I mentioned about impact patterns, where that blood source was in three-dimensional space. And so another reason why I say the requirements of the silver screen are not the same as for the crime scene and from a forensic blood substitute point of view is because we actually went in and did some research on it, and we created a series, a whole suite of simulations for typical types of blood stains that can be found at crime scenes, drip stains, for example, impact or spatter stains, for example, and even transfer stains. And we started making these series of validation tests. And so I don't have time to go through all the validation tests today, but I can explain in a variety of simulations to make these passive drip stains, for example. We created a 6-stage step that you can create these passive drip stains in a variety of different impact velocity conditions. I know this sounds way more physics than it is, but hang tight, and to make the blood stains of different size and morphology. And we actually went in and looked at some of these common blood substitutes and found out that while a lot of them can replicate the angle of impact, so we see uniform spreading in both planes, they don't actually meet the morphological parameters like size, spine, and scallop type, and number, and satellite spatter stains. These are satellite stains too here on the drip stains. And so color removed, if we're looking at blood substitutes or fake blood from common household items, this is what blood looks like. We can see that a lot of these household items and even some of these fake blood materials, glycerol based, for example, do not quite have the same type of morphology as you would predict or expect human blood to have at a crime scene. So we delved a little bit deeper. We suggested that, okay, maybe we should go back to the drawing board and start from scratch and look for designing blood substitutes. And so we gave ourselves 4 real design considerations, kind of like thinking about it from a chemical engineering point of view. We said that blood substitutes, forensic blood substitutes in particular, if these are going to act as standard reference materials, they should be safe and preferably free of biohazard risks. That's typically why you can't use them. They're validated in some sort of way, and particularly validated in the forensic context with the environmental conditions and a lot of common crime scene conditions that we would see. They're pretty easy to engineer. And bonus, they could offer some sort of advantageous feature for education or research and training. And so our first suite of blood substitutes were made using hybrid inorganic organic silicon sol gel chemistry. Why did we choose this? Well, we came from a lab that created high water content sol gels. These aren't your typical sol gels. They remain in the liquid phase for much longer. They have delayed gel transition. So you can simulate blood in the liquid form, and we were able to simulate kind of some of the drying mechanic features of blood stains across surfaces. So in a sense, in our world, we kind of showed that we made this kind of connection by suggesting that some of the blood stain mechanisms parallel deposition processes used in sol gel applications, drip coating, and things like that. For those of you that are new to the word sol gel chemistry, it's really a type of solution gelation chemistry. It's considered a two-step process where you form a colloidal suspension of solid particles we described as a sol, and then it's going to continue to polymerize to form a gel. So in our materials, the kind of base sol gel that we use before we add any sort of fillers to help with the visual appearance or any of the fluid properties of the material, we used a combination of tetraethyl orthosilicate and glycidoxypurple trimethoxysilane. They were hydrolyzed in a weak acid environment using a small amount of acetic acid, and I think it was like 0.05 molar or something like that. We see hydrolysis to silanols. They'll continue to polymerize and form some sort of colloid with a silica core decorated with epoxide functional groups that can be used for subsequent chemistries. And why we also like this is it was a nice kind of encapsulating agent for some of the biomolecules that we included in our materials. From a fluids and viscosity point of view, yes, congrats to those of you that checked viscosity on the poll. We found that a lot of these materials, particularly when we removed any of the residual alcohol or distilled these liquids, removed the alcohol from solution, we were able to mimic the non-Newtonian rheological properties of these materials. And in fact, we were able to show that these, particularly the rheology and the viscosity of the materials changed over time as well with blood. So that was great. We had a base material. Now can we add some fillers to it? Can we start getting into some physical simulations for blood stains? Can we do anything else chemically and maybe even biologically? Because these are all important bits of components of blood that are useful to a bloodstain pattern analyst. From a physical point of view, we were able to create a bunch of blood substitutes with a variety of fillers, like even simple fillers like magnesium silicate, for example, some dyes, some colors, some of the colors that you saw in the YouTube video before, and we were able to replicate the fine features. Notice we don't see a lot of spines in this case because this is quite a smooth surface, and the morphology of the bloodstain is dependent upon the surface upon which it is deposited on. We were able to do this in a lot of conditions. So we looked at making thousands of bloodstains in kind of commonly anticipated scenarios, different droplet volumes ranging from a few microliters up to 75 microliters, for example. Different dripping heights related to different impact velocities. And we were able to show that our materials fit within the expected range within these few hundreds of thousands of simulations compared to other commercial products and blood's kind of non-Newtonian counterpart, water, right, which is a Newtonian fluid. And we see these stains, they get much bigger and they don't have the fine scallop and spine features that you would see with blood and other non-Newtonian fluids. We also explored surface interactions, so congrats to those of you that checked that in the poll. And we were able to show that our blood substitutes did have similar surface interactions with common substrates to blood. Up top here on the image. And we did some fun things. We tried to simulate serum separation and some flaking too. It's not perfect. However, our group got into that world. And so we're creating quite the suite of materials, which is quite exciting. I want to stop and show you one more thing that's quite interesting about these blood substitutes from an experiential learning and STEM education point of view. Because we can now tailor these materials to contain lots of different types of fillers. These sol gels are pretty inert. We can tap into spatial reasoning of BPAs by exploiting concepts of color and how they can actually go in and process complex patterns. Because you can imagine at crime scenes, not a beautiful single impact pattern where one single drip stain is found. Unfortunately, you see pattern on top of pattern on top of pattern, and it's up to the BPA to interpret these patterns and figure out what happened in terms of the bloodshed. So for example, we made, we make a complex pattern. We go and we use this as this is our blood substitute. It looks a little orange on the slide here, but I promise you it's red like blood, okay? And so you say, okay, what happened? Okay, and rather than just telling the BPA, oh yes, I made an impact pattern and two cast-off patterns here, we can actually show them. So we added some cool fluorescent fillers into these blood substitutes, we dyed them in different colors, we made complex patterns, and now they can go in and see, oh yeah, I do see two cast-off patterns and an impact pattern, and they can start thinking about, you know, in real life, going back to say quote unquote real life, could I actually discern and see that there are actually two cast-offs? Can I not? What can I not? What's the limit of knowledge in this field? You don't believe me that this is this? All right, I'll show you one more. This is just the fluorescent FBS, just with a little flash in the photo too. Very cool. Love this part of the project, and in fact, we've used this training in a bunch of training programs around the world. So it doesn't stop out there. We can also do things like mimic the chemical enhancement properties of blood and diluted blood at crime scenes. So you can imagine that blood, so remember CSI, ooh, wow, okay, they spray luminol, you see the bright blue chemiluminescence. What happens there? For those of you that are new to the field, essentially heme acts as a catalyst in the oxidation of luminol in this reaction that promotes the bright blue emission, chemiluminescent emission of light. So we said to ourselves, okay, what's the easiest thing we can do with these sol gel systems in order to kind of exploit, say, a false positive reaction and make sure that it has this nice emission profile similar to blood. Notice, and one thing to note about blood with these emission profiles is that there's a slight delayed onset in the chemiluminescence, reaches a maximum emission intensity, and then it will slowly decay in terms of signal over time. So we thought, what can we do? Well, what's like hemoglobin? Horseradish peroxidases. So we were able to encapsulate horseradish peroxidate within our sol gel materials. We optimized the procedure, and by we, I mean Sumiko Polacco. This is part of her master's project in our lab a few years ago. And she optimized the horseradish peroxidase conditions, the solution conditions, the encapsulation conditions, and the sol gel matrix itself in order to develop a blood substitute that would give you similar emission profiles to whole blood. So she did this, and she did this really well, noticing that so here we're looking at, say, the chemiluminescence or the emission profile of the chemiluminescent reaction over time here. And she was able to create these FBSs, or forensic blood substitutes, that had horseradish peroxidase in them in order to simulate the reactivity with neat blood, so like whole blood, and also dilution profiles down to 1 in a million, because typically when you're spraying luminol or peroxidase reagents at crime scenes, you're not looking just for whole blood that can be visually seen with the eye. We're looking for latent evidence, so it's invisible to the eye, and so she was able to even mimic emission profiles similar to 1 in a million times dilution, which is a common dilution that you'll see in the forensic literature. What we found out too is that the sol gel matrix especially those with high water content, was able to encapsulate the horseradish peroxidase quite well, and we see a change. This is the neat horseradish peroxidase emission profile. We see that encapsulation in the matrix gives us that delayed onset and nice similar profile to human blood. We compared our materials to other commercial products, training liquids for training, and also controlled test cards to make sure that your luminol reagent is working at a crime scene. And we were able to statistically show, using KS, two-dimensional KS testing, that our materials had a better fit to the blood emission profiles than our commercial alternatives. And so we made a nice set of blood substitutes that could be used for training too. But in kind of similar light to the commercial too, we're able to create commercial standards that can be used just to spray to make sure that the reagent's working. So say you're a luminol solution, making up BPAs, making it up, make sure that it's working before they go spray down an area at a crime scene with chemicals. Can you see it's working or not? And so here's just an example of our blood substitute. It's put on a Q-tip. And then here's some screenshots of the chemiluminescence that we see over time in both our high intensity formula and our low intensity or low concentration formula too. So the next thing that our group is doing is we're not stopping there. We're trying to make some biological simulants too. And so we were able, so Amanda Orr is a PhD student in my lab right now, and she's working on immobilizing DNA within our sol gel and other types of matrices. And so we were able to do that too. You can imagine making kind of like simulating the white blood cells in whole blood, so that now we can make a suite of training materials that can be used to show can be used to simulate, say, genetic profiles, make sure the BPAs are swabbing the right areas, et cetera, et cetera, et cetera. But what we found out is that with these sol gel materials, they're great for physical and chemical simulation, but they're a little bit harsh on DNA, okay? Particularly if we're gonna start aminating or using aminated DNA to bind it to our colloids, we wanna make sure it's functional. It is functional, but we moved to other materials. So we started making hydrogel materials. These materials, we picked alginate type polymers, and we've cross-linked them using the classic calcium ion cross-links, and we've also covalently cross-linked our DNA into our material. So I'm really excited to talk a little bit about this project very briefly, because the paper just got published like last week, just about a week ago, and so we are able to make a variety of simulants that can contain DNA. So encapsulated DNA, and further work that Mandy's just finishing up right now is covalently cross-linked and dual cross-linked systems that have similar genetic profiles to human blood. We make profiles outside of the human range, so we don't have any issues, say, with contamination, et cetera, et cetera. But we make these blood substitutes, and again, they're to simulate ex vivo simulations. We know blood will degrade over time, these biological fluids, and so Mandy's doing a lot of work in looking at making these tissue simulants and how they can hold DNA and they degrade over time. What's interesting about this too is while they don't look like blood right now, we actually found that they have a lot of viscoelastic properties that are quite similar to a lot of tissue types that we'll see at crime scenes. So Mandy's starting to make a whole suite of tissue simulants, which is really exciting for our lab. And so here's just a little screenshot to show you that we do see changes in our elastic and even viscoelastic properties of our tissue simulants, and these hold true again over time and in various environmental conditions. So I know I'm coming close to my time limit, so I'll just tell you one quick thing and where we're heading next. So you can imagine we're continuing to build on our body of knowledge. One important thing to know about making blood simulants is that you have to understand the blood in the context that you care about it in order to go and mimic it. And so bringing it back full circle to understanding kind of the when, the time since deposition of blood, we're spending a lot of focus in our lab right now is just working with blood and figuring out a lot of these biomolecule and macromolecule degradation mechanics in order so that we get this information so that we can pick our best formulas or start making formulas that can actually start simulating the degradation processes of blood, or we can do it better, I guess, over time. So blood will undergo a bunch of physical changes over time, like gelation and desiccation. And we'll see a lot more chemical processes, even more than just the degradation of the heme or the hemoglobin protein in blood. We'll see things like cell lysis, VOC production, any type of biomolecule degradation, some metabolism, for example, and even some DNA, RNA degradation. Can we make blood substitutes that simulate that? So we're going all out on this area. We're really excited to make some blood substitutes that can be useful in the forensic context. The other added complexity to our research is that we always have to think about the applied. We're thinking about how do these rates of degradation processes change with the influence of the environment, whether it's photodecomposition or light, degradation pathways, and the influence of temperature and humidity on these processes. So it's becoming quite a complicated problem, but our group is up to the task. We're really excited about that. So in summary today, I just wanted to remind you kind of what I've talked about. And so at the end of the day, if you don't take anything from this lecture, just remember this: bloodstains and bloodstain patterns contain a wealth of information useful to a forensic scientist. It contains a wealth of biological, chemical, and physical information useful to a forensic scientist. And blood substitutes are still being developed. They're useful as forensic reference materials for ex vivo blood simulation. Forensic scientists cares a lot about that. We have shown in our group over the past a decade or so that silicon sol gel chemistry is useful in creating safe, robust, stable, and valid forensic blood substitutes. And now we're moving to the field of alginate-based biomaterials, and we're showing, at least we're hoping to demonstrate some promise in beyond blood simulants, but also in soft tissue simulants for forensic purposes. And so thank you so much for listening. A lot of this work wouldn't be possible without the most precious members in my lab group. So thank you, and also my collaborators and co-PIs and funding body NSERC. So thank you so much, and I'm happy to take any questions on the content that we presented today.
Okay, it's so exciting. That was such a great talk. Thank you so much.
Sorry about the technical issues.
Don't worry. So we have some great questions. Some people really came out with some great stuff. So I was trying to organize them, but there's so many, there's so many things that they ask, some in comparison to crime scene, some in comparison to actual blood itself. So I'm just gonna start with some earlier questions, and then we'll get into the more difficult ones. Is there a point that blood can be too old to analyze? And if so, how long is this?
Whew, that's a great question. And in fact, I mean, I don't think that there's a limit to what you can analyze in a blood stain over time. We've seen blood stains persist in, you know, I think I've seen historic cases with blood stains. I've also seen research that looks at, say, mRNA transcripts, for example, some DNA studies. You can still collect the viable DNA and RNA from bloodstains up to a few years post deposition. Some of the and obviously the heme will continue to degrade only to a certain point until really we see equilibrium with the hemichromes, for example, or that state. And again, it can be in the time scale of months to years to decades. And a lot of the time, environment pays a huge contribution to this. So, you know, up in Canada, we have very cold winters. You can imagine that the rate of degradation in bloodstains, and of course, crime scenes don't always happen inside. And of course, a bloodstain pattern analyst is going to be called to a crime scene in -40 post of fresh snow. And so sometimes they'll see that these bloodstains do persist, and in fact, the degradation rates are a lot slower in these conditions than, say, in the desert or in hot, dry environments as well.
Right. Oh my God, boy, do I know it. Crime scenes are very difficult to process, especially when certain weather conditions, and you kind of wish they were always inside, but you know, it definitely changes how you have to, you know, analyze the blood, analyze all of it, analyze the evidence, analyze everything. So, okay, so I'm gonna go into like a crime scene question. Okay. And, oh, well, since you brought it up, like what substances in an environment are actually going to affect how the blood spatter ages? Right. You know, is it something that could be like on the wall or something on the floor or something that, you know, the surface that it attaches to, that it's going to affect it?
Definitely. So first, it always comes down to how a bloodstain forms. And the way a bloodstain forms is really important in the way that it interacts with the surface. And surfaces can be non-porous, porous, semi-porous, for example, and that will dictate how a fluid spreads across a surface and also either adsorbs or absorbs into a surface. And so you can imagine some of those matrices are quite protective, and other ones we see, particularly hydrophobic surfaces, we don't see a lot of uniform, say, spreading. We see delamination of the bloodstain from that surface, and it will degrade in different rates. So the type of the surface is incredibly important from the point of the bloodstain formation to its continual degradation mechanisms. Also, we see that, like I mentioned, the environment plays a huge factor in the way a bloodstain degrades, forms and degrades at a crime scene. So things like temperature will influence some stain degradation mechanics. Humidity will. It will also really change the way a bloodstain is formed. And so I encourage you to look at, there's a lot of papers in the literature, particularly in the fluids literature, on how bloodstains form in different humidity conditions. And also, there are some biomolecules that will undergo some photodegradation. So light, and sunlight, indoor light, sunlight, for example, can change the degradation rates of some of the biomolecules in blood.
So now that you said that, that is such a great question, because do you feel that, or do you know, if there's any change in blood viscosity and color, like when the blood group varies? So if you have a different blood group, will the blood viscosity and color change?
Right. So I can take it back one step further and tell you that we have different fluid properties like viscosity, surface tension, density, and hematocrit between people. So we see interspecific variation in those fluid properties. And we also see intraspecific variation in these properties within yourself, depending upon what you ate, what activity you just did, any type of metabolic processes or whatever can all change your fluid properties, and these have direct implications in the bloodstain morphology. And so, we model, or a lot of physicists will model bloodstain formation using non-dimensional fluid numbers like the Weber number and the Reynolds number. And these are really dependent upon, say, combinations of inertial to surface tension forces and inertial to viscous forces. So yes, the viscosity is really important information, and we do see variability in blood stain size, shape, and morphology depending upon whose blood it is and what's going on with the physiology in that individual.
Okay, and then how about the addition of luminol? Like, will that affect the viscosity and the surface interaction?
Oh, sure. Oh, yeah. So I guess that's another area in kind of crime scene processing, is using chemical enhancement reagents to detect blood at crime scenes. So any type of dilution, especially with water-based reagents, can change the viscosity of a bloodstain. But I do want to mention, most of the time when these bloodstains form, they're now solids. So we're not really changing the viscosity of a liquid blood. We're talking about interactions now between a solid film and, say, the luminol solution on top of the bloodstain. Yeah, does that make some sense?
Yeah, okay. Yeah. I mean, there was a lot of questions, from the audience concerning luminol and then crime scene and then not disturbing the crime scene.
Right.
But they were, a lot of them were wondering what can prevent the luminol from working and like detecting blood, and that would cause it to give a false positive.
Right.
result.
Yeah. So there's a lot of reagents, like household reagents like bleach, for example. Even copper pennies can cause that oxidation of the luminol and cause that chemoluminescence. And so it's important to note for everyone in the audience, the luminol chemical enhancement test is a presumptive testing method. It's used mostly to see if there's any type of reactivity with the luminol reagent. If you see a reaction, it doesn't mean that it's blood. Okay, and so the BPAs are well versed in this knowledge, and they know that it's kind of like, it's like a searching technique to see, like, if I can't see any blood stains, and maybe there's a hypothesis that there are diluted stains, maybe there's a cleanup that's happened at a crime scene. Can you spray luminol in order to get some sort of reaction? And then you use that information into context. And again, it's important for the BPA to say that the limits of the technique. There's lots of false positive reactions for the luminol reaction, and so that they communicate that well with in the case context.
Yeah. So it's so funny that you say that, because someone had asked if we can affect the crime scene, but I don't want to touch that yet, because I want to ask you if they take, let's see, like, I don't know. Well, let me go back to this. I want to address this one first. Is there an actual product that causes the blood to be black when aging? when degrading, I
Oh,
should say.
yeah. I mean, this is it. It's like, what's the biomolecule? I mean, there's a suite
Yeah.
of so that paper that I was talking about on that slide there, check it out. It's by Zadora and Menzik. It's a one of the best literature reviews on the topic that I've seen. It's just recently published in Trends in Analytical Chemistry. And that discoloration is typically a result of the protein or the hemoglobin that's denaturing into a hemichrome state. Yeah.
Okay, so now I'm going to ask this one because I think this goes back to the luminol and your explanation of it. Can animal blood be easily or quickly detected from human blood? And I think that's a problem. Like when I teach personally, a lot of the students have a problem understanding, like, are we really concerned with animal blood? Right? Are we concerned with animal blood? Or are we only concerned with human blood? And I know that this comes up, and we got a few questions about it. So can animal blood be detected more easily from human blood?
Sure, yeah. And so first thing, if you spray luminol on animal blood, you're going to get a reaction too, because especially the mammalian blood contains hemoglobin and forms of heme, so you'll see that type of reaction. But as I mentioned at the beginning of the webinar, the BPA does have in their toolkit some techniques to say, is this blood probably human blood or not? And one example of this is the hemotrace test, and essentially it's like an antigen antibody test that screens for human hemoglobin. Now, we still call this a presumptive test because there are false positive reactions with some primates, and sometimes some, I think ferret blood, for example, can give a positive reaction to the hemotrace testing. But if the next thing you do then is swab the DNA and send it for genetic testing and look for human specific markers and hopefully be able to get a genetic profile from that blood stain, or human profile from that blood stain. And if it's not human, maybe it's an animal. Go and you can do some genetic analysis on that as well too.
Right. So one of our audience members would like to know, would brain blood be different, like identifiable wise, from stomach, like blood from a stomach wound?
Right. So at a bulk level, this is a good. These are almost stepping beyond my expertise. Remember, I'm a materials chemist that makes fake blood. But yeah, I mean, like a blood, depending on your presumptive testing reagents, they're not necessarily that specific. You'll still see a reaction. Maybe there are some brain-specific biomarkers. I'm not sure. I'd have to look into the literature if there are any specific metabolites that are found in blood from your brain versus other areas. But I know that there is a lot of research that does do, say, mass spec analysis and paired with statistical interpretations that are able to identify tissue types. So you can say this is brain matter, this is liver tissue, this is blood, for example. But I'm sorry, I don't know the full answer to that question.
It's a difficult one. I think these questions are amazing. Like they're so, I think they're great.
Have you ever incorporated this or encountered this in your job or your past job?
No. And unfortunately, a lot of my master's work was done in blood spatter, and my interest is there 100,000%. And that in autopsies. So when I dealt with autopsies, it was a lot. I didn't get to go to the scene. I got to see the other side of it. So to me, it was fascinating. I'm just fascinated by the way the human body reacts and everything that it does and everything that it encounters. And I think that using this blood and blood spatter to recreate a crime scene and figure out what happened, I think is absolutely astonishing, because it's just a matter of someone telling you a lie and you being able to go back and go, no, that's not what happened. This is what happened. And I just think it's phenomenal, and I love it. So unfortunately, I saw the other side of it, dealing with the bodies, and so I saw it completely separate from the crime scene, but all together it came beautifully, and it's funny that you say that also, because one of the questions that we got, and I love it because this is a question that I get in real life, how realistic is the show Dexter in comparison to real life blood analysis? Well, I want to watch the show, and then people started to tell me about it, and I was like, you know what, let me watch the show. So what do you think? Do you think it's realistic?
Okay, I want to see what you say after this, but I will preface that for those of you, maybe this is me showing my age. I'm more like Dexter in Dexter's Lab, not Dexter, the Dexter Morgan that everyone's come to fall in love with. But yeah, I mean, there are some recreations in the TV show that are, you know, slightly unrealistic. But that's really like any of the crime scene shows. They're meant for entertainment. And so, you know, sometimes there are some issues, and that's, you know, that it is what it is. And so that's all I'll say about that. What about you?
You know, I didn't watch it that much, and I was like, I can appreciate it just because I think they brought some information like to the public. Like a lot of people don't know about crime scene reconstruction. They have no idea what it's about. And they, you know, to see him do it and to see him stringing and going through the process, like that's some things that if you're able to do it with a classroom full of students, especially if you're an educator, it's amazing because they see the drama behind it. Like they don't just, you know, do a little test. They really get to evolve and understand like what we go through. You know, it's not something simple. It's not something that's done in 10 minutes. It's not CSI. It's reality. And so, you know, I love the learning process behind it. I think it's excellent. The show itself, like, I think it did a great job of just introducing that field to, you know, the public. And then
they showed too, these are scientists in a lab. Yeah, you said this way more eloquently than I did. So yeah, the introduction of CSI and Bloodstains was great. I mean, exactly.
Yeah. So
I mean, I answered the cool question. Are you like Dexter or like what?
Yeah, like when people say, you're a forensic scientist? Like, where do you, you know, what do you do? Like, you go to the crime scene? No, I'm the nerd in the lab coat sitting in the back. Like, and you know, and they have to understand that, like, we really are scientists that are doing, you know, quality work. Like, it's not just made up. And a lot of it's like, a lot of people think it's, you know, it's an applied science, which I understand, but you have to know the science theories and, you know, and you have to be skilled in order to be able to do all of this. So I think it's just, it's amazing, but
yeah,
I love blood spatter analysis.
Yeah,
so.
the added complexity to forensic science and knowing the science is being able to put it in an applied context, taking it from a pristine fume hood or a lab into the wild outdoors. It adds a complete amount, as like a massive amount of variation into your results. That needs to be accounted for using, you know, high-end statistical modeling, and it is complicated. And the other thing too, for the audience, that's you know, we take our work quite seriously because at the end of the day, a lot of this research or people doing casework, this comes down to you know, reporting on a criminal case, which someone's life and decisions around their life is on the line. So we take our science very seriously for sure.
Yeah, and I think that's the most important thing is that people have to understand that, like, we're applied to law, like we're forensics, you know, so at the end of the day, someone's life is on the line. And, you know, even, I don't want to say like dry labbing and all of that stuff that has happened in the past that people are reporting, you know, there are checkpoints that we have to go through and things that we have to, you know, we can't just go to a scene and just go to court and testify on things and like make things up. And I can definitely say there's definitely been some television shows on Netflix, and I won't say what they are, but you know, they're exposing what happens in this field. And I think it's excellent for people to see that, but they also have to understand, you know, where we're working and what we're doing is so important that at the end of the day, someone's life is, you know, in your hands. So you have to be extra secure in what you're doing and your analyses. And, you know, I think it's just super important. So from that Dexter talk, someone asked, and I think this is great, what's the lifetime of the blood substitutes like that you're using, especially when it comes to like in television, like what's the lifetime for these substitutes?
Yeah, great. That's a great question. Thanks for asking that. And like we saw in the YouTube video, I think it was like methylparaben or something was added to extend the shelf life of the
Right.
Hollywood type blood substitutes. Are those sol gel materials that I mentioned? They have I think I stopped counting after 2 years of shelf life, and they were still able to simulate some of the physical
Yeah.
properties. So they do have shelf lives of years just for the physical simulants. The chemical ones are a little bit at the moment. They have at least we tested. They have a shorter shelf life because as soon as that matrix solidifies, we see a decreased reactivity in the horseradish peroxidase, and so I think on the paper that we published and Smiko published in our research that I think the shelf life was somewhere around 6 weeks, and you could extend that shelf life if you put that product in a sealed and also in the refrigerator as well. So it is a feature that we care a lot about because you can imagine, shelf life is important, especially if you're doing training or you're doing bloodstain training in remote locations. Yeah, from a materials point of view, we definitely care a lot about the shelf life of our materials.
Is there any toxicity of the sol-based FBS?
That's also a great question. And so those materials are they're meant for bloodstain simulations, not on the body. So like making an impact pattern on a wall, for example, and doing those analyses here. And so we're moving into these biomaterials or bio-based systems in order to make sure that it's compatible, with simulations on human skin, et cetera, et cetera. So we're still working on it.
Wow. And your research, there was something that someone had asked. Would blood thinners or medications affect blood spatter?
Yeah, so I think there has been some research on looking at, say, the addition of warfarin, for example, in blood. I can't fully remember the conclusions of the paper, but it may fall within the variability of kind of like the human population in terms of like the spread.
Yeah. So does like the neutrality of the manufactured blood, does it matter in comparison to like real blood? Like if
Yeah. Well, we made our systems. They have varying pHs. Some are a lot more acidic than human blood, and some systems do fall within neutral pH. Again, sometimes we optimize those conditions in order to simulate one specific mechanism about blood. So in a perfect world, we'd have a Cadillac material that's, simulates a lot of different properties of blood. We're still getting there, but pH is definitely important.
Okay. All right. So I think, let's see. I have this great question and I really want to save it for the end, but I think it's important now because it might tie in some of the other topics that people had. This question from audience member, absolutely love. What does the future of technology, like where does it lie? 10 years, 50 years, 100 years? Could synthetic blood ever advance enough to substitute for real blood in medical procedures?
Great question. That's a great question. And so note, I'm not an expert in medical blood substitutes, but I do know that they are right now, they're pretty good. There's some, I think there's from a kind of bird's eye point of view, looking at the literature, sometimes there's some issues with the oxygen transport, and some of these compounds actually really hold on, have a high affinity for oxygen, and have some issues releasing, but I could be wrong. But yeah, sure. I mean, the sky's the limit. The more people that get into, and I'm the eternal optimist researcher, the more people that get into the field, why can't we start trying to make materials that could be more and more realistic? I mean, it does take a lot of time. A lot of our research, or where the time in our research, we put a lot of time and effort in developing validation studies because we want to make sure that these materials are behaving like blood does outside the body. So yeah, maybe. I mean, are we there yet? No, but that's great. You know, this is why I'm in my job. And in terms of technologies in the future, oh man, with there are so many different technologies that can be useful to a bloodstain pattern analyst, particularly 3D laser scanning. That's more kind of like on pattern analysis side. From a chemical point of view, you know, we're really interested in mass spec imaging. You know, can we do a total profile and a total map of a blood stain, and what type of information can we get from that? There's always improvements in genetics, and you know, especially in sensitivity in these equipment are incredible, and so that opens up a lot of doors for ultra trace evidence analysis and maybe even cold cases as well.
That would be amazing, especially with the cold cases, because there are so many cold cases from so many places and years that just have not been solved. I mean, we know definitely some famous ones, but I wish technology could get to the point, and I hope and I think that it will get to the point that we will be able to solve them. I mean, it might be a little late, but at least, you know, they're solved and they put an end and close it, really, and close the case, bring some people some sense of relief in that sense. But
yeah,
okay, so since you said the crime scene stuff, there's a couple questions we're gonna go before the end. I just want to ask this one. I think this is important as far as like crime scene reconstruction is concerned, but one of the audience members asked, does the radius of the spines around the droplet tell you more about the height it was dropped from or the surface that it fell on?
Great question. In laboratory studies, there are studies that indicate that you can use spines and to get some sort of information about how high a blood source was before it was dripped on a surface, so like that height. But that's really in lab conditions where you know the exact fluid properties of the blood, and you are working with pristine surfaces. And so if there's any type of point defect in the surface, or maybe there's the blood source has a slightly different surface tension than normal, or the average, then we will see differences in numbers of spines and their morphology. So in theory, there's a lot of really nice research that demonstrates the utility of the technique, but in the real world, it's quite a variable process, and so that's why it's not really used in order to give height estimates and things like that.
Okay, so have you tested, this is a great question, how spatter patterns produced by FBSs compare to patterns produced by human blood, and specifically determining the region of origin?
Yeah, it looks like we have a bloodstain person in the audience, so that's exciting. Hello. Yep. Yeah, we do have some research and some validation studies on impact patterns analysis, and so I think you'll have to dig into the literature on that one. I think it was published in the Canadian Society of Forensic Science journal, but we have done some validation studies on that too.
Wow. Okay. And as a forensic scientist, how do you deal with the potential emotional aspects associated with analyzing a crime scene?
Great question.
Yeah.
And no, Monica, we'll hear from you as well. So I'm mostly a research scientist. So at the moment, I'm not going to crime scenes and I'm not doing casework. I'm in the lab doing research. But it's definitely a serious topic, and there are ways to make sure to analyze crime scenes, making sure that you know, you're looking at the evidence objectively, so that you know you don't go down any rabbit holes, or you know, sometimes there are emotional you know, results from going into crime scenes. It happens, but the important bit is being objective in your analysis, going in and doing what you came for. And so again, but I can't really speak so much to that because I'm a researcher in a lab. But Monica, I think maybe you can with your crime scene experience.
Yeah, I think it's hard. It's something that I tell my students that when you enter this field, it is difficult. You have to be able to separate them. So not only do you have to, analyze the evidence and be objective and put that to the side. You have to put your emotions to the side. You cannot let them get involved. Like, you really have to shut down. And it's really difficult to do if you're not used to that. I mean, especially if you see children at a crime scene, or I know me personally, like performing autopsies on a kid or someone my age. It really like puts your life in perspective. So it is difficult to do. And that's something that I say to them. I also say to them, like, if you can't testify and get up in front of people and be able to, hold your composure while someone is ripping you apart, that's another thing, that you really have to think about when you go into this field. So it is difficult, but if you can separate it out, and that comes with experience, I think everyone crosses that bridge at some point when they're in the field. You see something that shocks you, or you see something that hits you emotionally, you're like, and then you just have to walk away for 2 seconds and then come back, pull yourself together and go forward with your analyses. So there's no time, there's just no time to get emotional. You just have to do what you need to do, move forward. So yeah, so I think we have time for one last question. So I just want to ask you, what is like one important lesson that you want to leave with the viewers? I mean, this was a great talk.
Thanks. Well, thanks for having me. I've really enjoyed this. And if there's one take-home message, it's really that blood is, blood, the aqua vitae of life and death. It's a very complicated fluid and material. And in order to make a blood substitute, you have to first understand how it behaves in the context that you're interested in.
All right. Thank you so much. This was so much fun. And I just want to pass it off. I just want to thank everyone for coming today. This was excellent. It was great working with you.
Yeah, thanks so much. I really enjoyed.
Thank you.
Thank you so much, Theresa and Monica. Here we go. And sharing your knowledge about forensic chemistry and the exciting future of FBSs. If you enjoyed today's topic, check out the short video content from our ACS co-producers, Reactions. In the Death and Decomposition episode, discover how humans may be breaking the cycle of life because of our burial practices. Before we sign off, we would like to invite everyone to these free upcoming broadcasts. How is lab productivity often strongly linked to good lab housekeeping? On Thursday, November 4th, discover a proven approach from leaders of graduate research lab safety teams that aims to promote research productivity. How do you match your personal skills with the job requirements in order to thrive in an ever-changing job market? Join us on November 10th for a special broadcast for those that reside in India to learn about choosing a career based on personal strengths and values. Also, on November 10th, we're having a conversation with industry experts on building sustainability into every innovation from the ground up. Learn about the future of sustainable innovations being developed in corporate research and development. We'd love to hear what you think about today's program. You can reach out to us through the ACS social media channels on Facebook, Twitter, and LinkedIn, or by email at acswebinars@acs.org. We're curious to hear what you thought of today's broadcast. A short survey will pop up directly after the webinar that will take only a minute to complete. Also, remember that an invitation email to view the unedited recording from today's presentation will be sent to all registrants for a 24-hour period, after which it will be removed, edited, and will become an ACS member benefit. And that wraps up our program for today. On behalf of all of us at ACS, thank you for joining us. We hope to see you again soon. And until then, happy haunting, my creepy chemist friends.